Okay, good afternoon, everybody. My name is Paige Beliveau, and I am here with the CHAMPS team. I'll be facilitating this webinar this afternoon. Before I hand it over to Lars, I just want to go over a few housekeeping things. If you are in a noisy area, or even if you think you aren't in a noisy area, we just ask that you put yourself on mute so that any feedback coming from your phone line doesn't interfere with today's presentation. Please don't press this on hold, just in case you have hold music set up. That would also be a bit distracting. If you have questions, we'll try to save some time for the end. We ask that you please type them out in the chat box so that as we're going along, if you have a question, you can type it out while it's on your mind, and then at the end we'll read them out loud and give Lars a chance to answer them. Can we go to the next slide, please? One more thing is that we are going to be doing a 4-hour clinical skills training at Anderson on February 20th. We still have spots open, so if any of you hospitals need staff trained, we encourage you to go sign up by logging onto our website at that link there. Kathy Carruthers will be leading this one, and we know how much you guys appreciate seeing her training. So once again, just check that out on our website. At this time, I'm going to hand it over to Lars, who will be our presenter today.
All right. Good morning or good early afternoon, depending on where you are in the U.S. We'll be talking about human milk oligosaccharides today. Thank you very much for the invitation and everyone joining and for your interest. Usually I ask the question, who knows about human milk oligosaccharides, and then I get the arms up, but I guess on a webinar that's not possible. So I just dive right in with first disclosures. So this is how my research lab and our center is funded currently. And also a very important disclosure that none of the work really would be possible without amazing lab members and really collaborators from the US and around the world. We have a very extensive network of collaborators really on all continents. And none of what I'm going to present today would be possible without any of their support. All right, so I'm starting with a bold statement here that I think most of you probably have seen already. It's from the editorial in the Lancet Breastfeeding Series 2016 that says that the death of 823,000 children and 20,000 mothers each year could be averted through universal breastfeeding, along with economic savings of $300 billion US. And while that is a bold statement, really it raises a few questions. Why is that? What is in human milk that makes it so powerful? And how does this all work? Like, what's the mechanisms behind that? So let's lift the curtain of this statement here and see what is in human milk and what's so unique about human milk that makes it this powerful. So if you just look at the macromolecules in human milk, you see there's protein, fat, and lactose, but there's also oligosaccharides, 5 to 15 grams per liter, which often exceeds the total amount of proteins. If you compare that to bovine milk, there's only about 100 to 1,000-fold less of these oligosaccharides. The structures are less complex, and since bovine milk is the basis for most of our infant formula, there's hardly any oligosaccharides in infant formula compared to this huge amount of complex structures that we find in breast milk. Now, what are human milk oligosaccharides, or short HMOs? They are complex sugars, not just quite like the sugars that you might have in the coffee or tea in front of you right now. That is fairly complex already if you look at it from a chemical structure, but human milk oligosaccharides are even more complex than that. So if you look at the chemical structure here of a fairly simple human milk oligosaccharides, you might hit the, not just the mute button, but just get off the call, because it does look fairly complicated, even if you are a chemist. So we make our life a little bit easier here and give each building block, each monosaccharide, a different symbol, and then assemble those symbols and those building blocks here in different ways, and that makes it much easier to talk about oligosaccharides that way. So you see there's really only 5 building blocks. There's glucose and galactose, which you might know from lactose. And then in addition to that, there is N-acetylglucosamine, fucose, and sialic acid. So this slide here is going to show you what we call the HMO blueprint in this blue box up here. And then I give you a few examples of how different oligosaccharides look like in the white space that is around it. And again, here's our 5 building blocks. And you can think of that as Lego bricks that we put together in different ways. And then either you put them together to get a house or a car or a train or a castle or whatever you like to build with Lego. So again, all human milk oligosaccharides here have this lactose, glucose, and galactose at the reducing end, and then it's built on that. So these are free sugars not connected to proteins or lipids. And we always add these disaccharide units to extend human milk oligosaccharides. So you can do that in different linkages. You can branch out the structures. You can do that multiple times and get fairly complex backbones. And then in addition to extensions and branching, you can also modify the oligosaccharides with either fucose in different linkages. If you add fucose to lactose itself, you get 2-fucosylactose and 3-fucosylactose. And specifically, this 2-fucosylactose you might have seen because it's currently added to some of the infant formula products. So it says on the package contains HMOs, and then in small letters it says contains 2-fucosylactose. You can add fucose also to the growing chain, and if you look at these 4 oligosaccharides here on the bottom, if you counted the building blocks, they are identical between these 4 oligosaccharides, but they're just put together slightly different. We call that structural isomers, and these structural isomers, we believe, have different functions. So depending on how you put the monosaccharides together, you get different functional units. Then it gets a bit more complicated from there. You can also add sialic acid, which is this diamond here, this purple diamond, in different linkages. And sialic acid introduces a negative charge to your oligosaccharides. If you add sialic acid to lactose, you get 3 and 6-sial lactose. And again, you can add it also to the growing chain. You get different structural isomers, these 3 down here. You can add more than 1 sialic acid, and you get, in this case, 2 negative charges on the molecule. And you can continue this on. And so far, we have about 150 to 200 different oligosaccharides identified by structure. Now, the really interesting part here is that every mom makes a different set of oligosaccharides in different concentrations. Here is an example of about 1,200 samples from the Canadian child cohort, and you see different subjects on the x-axis, and then the different concentrations on the y-axis, different colors being different oligosaccharides, and you can see there's some milk samples here with fairly low concentrations, and then on the right you have some milk samples with very high concentrations. And if you take that as relative composition, so you set it all to 100%, you see that within that there's huge variation in how different oligosaccharides contribute to the total amount of oligosaccharides. So it's almost like a thumbprint that I show up in the corner there, a milk drop with a thumb in it. It's almost like a thumbprint that every mom makes a distinct set of oligosaccharides in different concentrations. Now, I said earlier that bovine milk barely contains any oligosaccharides, and the structures are different. So that means that infant formula also contains different oligosaccharides, and infant formula companies have tried to mimic that by adding different oligosaccharides to their products. 2 examples here are galacto-oligosaccharides, GOS, and fructo-oligosaccharides, FOS. But you see GOS contains this multimers of galactose that we don't really find in human milk, and FOS contains fructose, which is not present in human milk as well. So again, comparing that to human milk oligosaccharides, those artificial oligosaccharides are structurally very different from human milk oligosaccharides. But some of the products now do contain first individual human milk oligosaccharides, so 2-fucosylactose being an example. Now, that is what oligosaccharides are. What really happens to them when they get ingested with the milk? So baby takes them up. They get to the stomach where they're very resistant to the low pH. They're not digested by pancreatic and brush border enzymes either. About 1% is absorbed intact, appears in the urine, and then the rest is either degraded in the colon by bacteria or is excreted intact with the feces. You see this fairly big range here, 10 to 80% are excreted. Now, what is it now, 10% or 80%? That's almost saying all or nothing, 0 to 100. It depends on the time of lactation. So very early on, in the very first few weeks, when the bacteria in the intestine are not fully developed yet, most of the oligosaccharides go through. Then the bacteria start to metabolize oligosaccharides and take use of them, and then less and less oligosaccharides are excreted with the feces as the gut microbiome matures. All right, let me summarize this very first part on what human milk oligosaccharides are so we're all on the same page here. They are the third most abundant component in human milk. The composition follows a basic blueprint. There's more than 150 different oligosaccharides identified so far, and they vary between different moms and to a certain extent also over the course of lactation. I'm going to come back to that in a minute. Infant formula oligosaccharides, the GOS and the FOS, are structurally and functionally different from human milk oligosaccharides. Human milk oligosaccharides reach the distal GI tract intact, but they're also absorbed and reach systemic circulation and organs other than the gut. So when we talk about the function of oligosaccharides, we focus mainly on the GI tract, on the intestine, but really there's also potential functions outside of the intestine. So we talk about this variation in human milk oligosaccharide composition, a thumbprint, different from mom to mom, but what really drives that variation in HMO composition? So what are the maternal drivers? And the first part that I'd like to talk about is genetics. I think that is very interesting because it's really the main driver of variation, and it really makes it a black and white difference. If you take a look at this graph here, this shows you what we call a principal component analysis. Every dot on this graph here is an individual human milk sample from an individual woman. The closer the dots are together to each other, the more similar are the oligosaccharide compositions in those two milk samples. The further apart those dots are, the more dissimilar, different are the oligosaccharide composition in those samples. So if I highlight that here with the pointer, you see this sample up here, for example, the oligosaccharide composition is very different from the sample down here. On the other hand, these samples that are very close together, oligosaccharide composition is very similar. Now you see already that I highlighted two different colors here. Oops, what happened? That I highlighted two different colors here. There is this cluster on the left, and then there's this cluster on the right, and there seems to be some space in between. So there's something driving that differentiation, that some moms are more in the left cluster and some moms are more in the right cluster. And we can ask what maternal factor really drives that difference. And if we just look at this in the child cohort, in the Canadian child cohort that are highlighted here in bigger bubbles, each sample, that separation can be explained by one single nucleotide polymorphism. So it's one base in the DNA that's different. And I explain to you in a minute what that does and why that drives this huge variation. So keep that in mind. It's one single nucleotide polymorphism of one SNP that drives this variation. So back in the child cohort here, we looked at associations between oligosaccharides and how that associates with different SNPs in different genes on different chromosomes. And you see here there's something on chromosome 19 with this highly significant. So this is your p-value here, and that means there's 80 zeros before you reach the 1. So highly significant associations between different SNPs and oligosaccharide composition, in this case, 2-fucosyl-lactose. And it turns out that this area of the chromosome 19 encodes a protein called FUT2. What is FUT2? What does that to do with oligosaccharides? FUT2 stands for fucosyl transferase, so it transfers fucose onto underlying structures. And you see here is your FUT2 enzyme, and it catalyzes this reaction down here to add fucose in this alpha-1,2 linkage to the terminal galactose. There is another fucosyl transferase here, FUT3, that catalyzes the reaction of adding fucose in an alpha-1,4, alpha-1,3 linkage. So if you have a SNP, one base pair exchange in this enzyme here, it actually introduces a stop codon. The enzyme is no longer functioning, and the enzyme is dead, and you get oligosaccharides that lack this specific structure up here. So these moms can no longer make this alpha-1,2 linked fucose. They still have an active FUT3. They can still make other oligosaccharides. Then there's moms that have an active FUT3, but there's also SNPs in FUT3, and they can't make these structures over here. And then there's about 1% of the population that have inactive FUT2s and inactive FUT3s, and their fucosylation pattern on oligosaccharides is very different, very limited. So just by having those two enzymes and just these two SNPs makes the oligosaccharide composition extremely different. Here's an example how that looks like in the lab. These are two different chromatograms from oligosaccharide composition. We use this peak 1 here as our internal standard, so you see that's the same. So we can compare those two samples. Here's a sample from a secretor mom. She has an active FUT2. This peak number 2 here is the dominant oligosaccharide, and that happens to be 2-fucosylactose. So it has this alpha-1,2 linkage. The sample over here on the right is from a mom that is a non-secretor. She can't make FUT2, and peak number 2 here is completely absent. She cannot make these oligosaccharides that contain alpha-1,2 linked fucose. Now, that all sounds very chemical and analytical. Why is this even relevant? You can imagine that if we find that these oligosaccharides that contain these structures have a benefit for the infant, then some milk samples might confer that benefit, and other milk samples might not. And most intriguingly, it's not in here yet. So here's really how this looks like. It doesn't only affect this 1 or 2 oligosaccharides. So here's just this one SNP that we call the Caucasian SNP because it was mostly identified in Europeans, but there's other SNPs in other populations. And you see there is no 2-fucosylactose in the ones that have this SNP. There's also other oligosaccharides that are not there. Then other oligosaccharides are higher expressed. It affects oligosaccharides that are not fucosylated. And it affects oligosaccharides that are sialylated. So it's not just the oligosaccharides that are directly impacted here. And in fact, it's the entire sum of oligosaccharides that's lower when moms have this SNP. Fucosylation is impaired, but sialylation goes up. Okay, so back to why this is important. We actually see that the ratio between secretor moms and non-secretor moms is very different depending on where you look. So here, for example, on the Latin American continent, in South America, you find in Peru, most moms are secretors. And there's only 1 or 2% of the moms that are non-secretors. And you would wonder, why is that evolution has pushed these moms to make these specific oligosaccharides that are secretor dependent, whereas if you look at other parts of the world, there can be more than 30%. For example, here in Northern America or on the African continent, more than 30% of moms are non-secretors. So there must have been some benefit of mom being non-secretor to mom being secretor. So there's some different evolutionary forces that drive human milk oligosaccharide composition in different areas of the world. Okay, let's skip this one. So that's the genetic part of it, and that really drives the black and white difference how oligosaccharide composition looks like in different moms. I think this is probably the strongest driver of oligosaccharide composition variation. There's other genes that are involved in that give more subtle differences, and we're trying to puzzle this all together. You'll be surprised that if you open a biochemistry textbook, the synthesis of human milk oligosaccharides in the mammary gland is not fully understood. So there's still a white page that needs to be filled. And of course, it's something that's related to only women and only during lactation that we haven't fully understood yet. I think that's very typical of the field that knowledge is quite limited in that area. So that's genetic factors, but what about environmental factors? What about diet? Can we change maternal diet to drive oligosaccharide composition? While we don't have good data in humans, we can do these studies in animals. Most mammals or all mammals really, make oligosaccharides, different ones from humans. Mice, for example, that we have in the lab, make only 2 oligosaccharides, 3-sialactose and a little bit of 6-sialactose. And we can milk a mouse and see what the oligosaccharides look like and how much is in each milk sample. So if you take a mouse under normal food conditions in a regular cage, you can measure 3-sialactose concentration in the milk, and then you can, for example, put a mouse on a high-fat diet. So on a high-fat diet, the 3-sialactose concentrations are reduced. So that's the first hint, really, that diet might change oligosaccharide composition. Then if you exercise an animal, not with weights, but on a treadmill, you would see that the oligosaccharide concentrations actually go up. And then if you take a mouse on a high-fat diet and train it, you see that the oligosaccharide concentrations come back down to almost normal levels. So food intake and training really have a compensatory effect here. Well, that's great. That's animals, and how relevant is that for what we do in the lab? I think it does give a good hint that there might be something going on in humans, and we have some first data that shows that that might actually translate to humans as well. So here we've done a study in collaboration with a group at Baylor where we take moms and we switch them over in their own diet so they become their own controls. We either switch them from a high-carb to a high-fat diet, or we switch them from a high-glucose to a high-galactose diet. And the switch to a high-fat diet shows you that, indeed, thiolated oligosaccharides are significantly lower, just like we see it in the animals as well. And that does not happen for fucosylated oligosaccharides, but fucosylated oligosaccharides go up when you switch mom from a high-glucose to a high-galactose diet. So very low numbers, very preliminary data, but a first hint that maternal diet and potentially exercise as well have an effect on oligosaccharide composition. Here's a paper that we just published a couple of weeks ago where we conducted a randomized controlled trial with probiotics during pregnancy and then looked at oligosaccharide composition in the milk during lactation. And you see that there is indeed some differences. So far we thought that oligosaccharides shape microbial communities, but it could very well be that microbial communities also shape oligosaccharide composition. So it's not just a one-way street, it's really a two-way street the way it's depicted here on this cartoon. So there's a few things that we can do with diet, with supplements to change oligosaccharide composition. And physical activity might be also one way. What about exposures? What about exposures to, let's say, smoking, medical drugs, recreational drugs? How does that affect milk composition, specifically looking at oligosaccharides? And the answer is we don't know. There is no data at this point to point us in the direction whether any of these exposures influence oligosaccharide composition. What about health and disease, maternal health and disease? What if mom has a gestational diabetes, chronic inflammatory disease, you name it? What is the impact on oligosaccharide composition? Again, we have no data on this at all. We have no data on how maternal disease states could potentially affect oligosaccharide composition. Now, what about time? I said earlier that oligosaccharides change a little bit over the course of lactation, and that is shown here again from the child cohort, where you see specifically now all oligosaccharides listed here. You see for 3-sial lactose, the oligosaccharide concentration goes up over the course of lactation. 6-sial lactose, the structural isomer, the concentration goes down. DSLNT, concentration goes up. And you see really there's different oligosaccharides that change over the course of lactation. Very interesting also, not only over the course of lactation, but timing also is relevant when it comes to the season of collection. You see there is some seasonal patterns where oligosaccharides are different in the spring, for example, than they are in the fall. And that is very interesting to see. And we've seen this in this cohort, and we've seen it in a cohort in Gambia as well, in Africa. And we don't really know how that works. We thought originally that it is potentially vitamin D exposure. And we've seen that in 2 intervention studies now that might not be the case. But there's something to seasonability here that changes or impacts oligosaccharide composition. So what about parity? What if it's the first child or mom already had several babies before? Does that have an impact? And you see here on the right that at least in the secretor moms where we have a higher N, we found that there is a higher concentration of total oligosaccharides if mom already had 1 or 2 children before. That could potentially be related to maternal age. You can imagine the more infants or the more babies you have, the older you are probably. But we looked at maternal age, and there is no significant difference in oligosaccharide composition. And you see down here mode of delivery, whether it's vaginal delivery or cesarean delivery, there is no difference in oligosaccharides either. All right, so that is some of the maternal factors that drive human milk oligosaccharide composition. You see there's a lot of question marks really still on how this all works and how this all comes together. But again, that triggers the question, why is that even important? Why do we need to change an oligosaccharide composition one way or the other? What's the benefit really? And that brings it to, what are the infant outcomes? Like, why is this important? What are oligosaccharides actually doing? And I just put together a laundry list here of what contacts human milk oligosaccharides are studied right now, and I'll give you a couple of examples later on. But this is really a laundry list of all the things that we think oligosaccharides could potentially contribute to. And the list is probably open still on the bottom, so we can probably still add a few things that we haven't thought of yet that will come up over the course of new discoveries. But this is really what the list looks like right now. And let me show you real quick what we think oligosaccharides are doing in a cartoon, and then I give you a couple of examples of real-world science that we've done to show that oligosaccharides are actually doing something. So this cartoon shows you the intestinal lumen. You have intestinal epithelial cells here on the bottom. You have potential good bacteria here in green, potential pathogens in purple, and then our oligosaccharides on the top. And I'm going to show you in the next couple of minutes how we think those different factors interact with each other and how that might relate to infant health and development. So for example, we know that oligosaccharides are prebiotics. So they have some beneficial effects on the potential good bacteria. These bacteria are able to metabolize oligosaccharides, use them as a growth substrate. They have an advantage over other bacteria that cannot metabolize oligosaccharides and really fill that niche and kick out the potential bad bacteria. We've worked here with Karsten Zengler just one floor down to see, well, there is 150, 200 different oligosaccharides. Do different structures have different effects on microbial communities? And the answer is yes. So the prebiotic effects of oligosaccharides are structure specific. It depends on what oligosaccharides you feed a microbial community, and they develop very differently. And I think that's important as we start to look at how different milks affect microbial communities, but it's also very important when we start to introduce individual oligosaccharides to infant formula and say that adding one or two oligosaccharides have the same effect as adding the full mixture of 150 or 200 different oligosaccharides. So this might have short and long-term consequences for infant health and development. In addition to just being food for bugs as prebiotics, oligosaccharides also have effects on the bad guys directly, so they are antimicrobials. I'll give you one example here that we've done with Victor Nizet at UCSD, where his postdocs sprinkled oligosaccharides on a variety of different pathogens that they had grown in the lab, and one of them was group B streptococcus. And group B streptococcus grows nicely in the lab without oligosaccharides, but it was very interesting to see that once you introduce human milk oligosaccharides, group B streptococcus would just stop growing. It would just not grow anymore. And we have identified the structure that's responsible for that. So again, it's structure specific. It's individual oligosaccharides. Not every oligosaccharide has the same effect. We identified the target on group B streptococcus that we think is targeted by the oligosaccharides. And we think that it really acts as a inhibitor of growth by blocking cell wall assembly. This is one example. We just happened to test these 4 pathogens. It could be very relevant to other pathogens as well, whether it's bacteria or protozoan parasites or other pathogens. So very interesting for this project really is that once we introduced common antibiotics and did a combination approach of antibiotics and human milk oligosaccharides, we found that there is some true synergy. So you would need less antibiotics in the presence of human milk oligosaccharides, so really lowering the dose of your antibiotics to have the same effect. And that, of course, is very important as we talk about antibiotic resistance that's emerging in hospitals and really around the world being a major crisis.
Lars, if I can interrupt you for a second, we've lost visual on your presentation. Are you able to re-upload that?
Yeah, I can see everything still. Let me just put this together here. When did you lose visual?
Maybe 2 minutes ago. I'm assuming just the internet connection.
No, I did. I printed this, but now I'm done.
Oh, let's see.
Yeah, I'm good. Thank you.
Okay, can you see it now?
Yeah, there we go.
Okay.
Thank you.
So here we go. Okay, thanks for letting me know. So in addition, we talked about prebiotics, we talked about antimicrobials and potential for the emerging crisis of antibiotic resistance. Now there's a third factor how human milk oligosaccharides could potentially affect this interplay between host and microbe, and that is by serving at what we call as anti-adhesives. So let me go one step back here, and we have now in this cartoon a pathogen exposing or approaching the intestinal epithelial cell surface, and you can imagine if you're a pathogen in the intestine, there's a lot of movement going on, and really the intestine tries to move out and move everything that is in the intestine forward to eventually come out. So if you're a pathogen or bacteria in general, you somehow want to hold on to the surface to not be washed out and not be able to colonize. So very often the way that happens is that bacteria have what we call lectins or adhesion molecules that specifically bind to sugar molecules on the epithelial surface. And from your anatomy class or physiology class, you might remember that we call this the glycocalyx. So it's this sugar coat that sits on our epithelial cells. It's fairly thick, actually. And bacteria really try to bind to these sugar molecules on your epithelial cells. Now, it turns out that's very important for bacteria to proliferate and in some cases to invade and cause disease. And if we go one step back and now look at the situation in the presence of breastfeeding, so when human milk oligosaccharides are present and they're coming in huge amounts every 2 to 3 hours and flush the intestine, human milk oligosaccharides are very similar in their structure to those structures that we have on our epithelial cells. So human milk oligosaccharides now serve as soluble decoy receptors and block bacteria from binding to the epithelial cells, and in the presence of oligosaccharides, bacteria get washed out without being able to attach and without being able to cause disease. That has been shown for Campylobacter, for example. We've recently shown this for enteropathogenic E. coli, EPAC, that in tissue culture you can block adhesion of EPAC to intestinal epithelial cells or epithelial cells in general. And not only that, EPAC is known to cause lesions on epithelial cells, so it really starts to destruct the epithelial cells, your inner lining of the intestine, and that's depicted here, these light green spots on the cells. That's where the lesions are formed. That's where damage is happening. And you can see the amount of lesions are much, much lower and really significantly lower if you quantify it when you treat the cells also with oligosaccharides. So that's the anti-adhesive effects. Then there's also direct effects on epithelial cells. Human milk oligosaccharides can change the programming of epithelial cells and the response of epithelial cells. And this might backfire into the lumen. There's antimicrobial peptides that are secreted and that change the microbial communities in the intestine. We'll skip this one. And then there's also effects of oligosaccharides on immune cells, either underlying the intestinal epithelial cells or systemic immune cells, because if you remember that from earlier, human milk oligosaccharides are absorbed intact and make it into the systemic circulation. So they are there in concentrations relevant to modulate immune cell responses as well. And that's something we're doing with Philip Quartz here, where we look specifically at chronic macrophage inflammation. All right, so summarizing this part, what are the potential benefits of human milk oligosaccharides? How does this all work? Well, there's indirect effects on the infant through effects on the microbiome or microbial communities. Oligosaccharides are prebiotics, antimicrobials, and anti-adhesives. But there's also direct effects on the infant independent of the microbiome by changing epithelial cell responses, immune cell responses, and multiple other mechanisms that we haven't really discovered yet. And this might have health implications throughout the entire life cycle. And I show you that on the next slide, what I mean by that. When we think of effects of breastfeeding, we really think of the immediate short-term effects first, and in this case, on the baby while it is breastfed. But really, we also know that whatever happens during the first few months or years of life have long-lasting effects for later on in life. So this really affects the entire lifespan all the way up to the elderly. We've recently shown that we find oligosaccharides we find oligosaccharides in the systemic circulation of breastfeeding women as well. So there might be effects on the breastfeeding mother as well, not just on the infant. And in fact, we find these oligosaccharides already at the end of the first trimester during pregnancy. So there might be effects on mom during pregnancy already. And again, this might have long-lasting effects for later on in life as well. And since it is already in the systemic circulation during pregnancy, we've recently shown that even the fetus is already exposed to milk oligosaccharides in utero. We found oligosaccharides in amniotic fluid, and the question is, what's the immediate effect on the growing fetus, and what's the long-lasting effects of that exposure? And then there's individual oligosaccharides, synthetic oligosaccharides now available that we could use as therapeutics potentially really in all stages of the life cycle. And we're especially interested in the elderly. How can we use oligosaccharides as novel therapeutics that are safe and that are battle tested during evolution really to be effective as well? I give you a couple of examples, different examples. One is the immediate short-term effect, and that is oligosaccharides in the context of necrotizing enterocolitis. That's some work that we've done over the last 10 years. What is necrotizing enterocolitis? Still one of the most common and fatal GI disorders in preterm infants. About 5% of all very low birth weight infants develop necrotizing enterocolitis, and about a quarter of them die from the disease. And we really got interested in that because of the observation that there's a 6 to 10-fold higher risk in formula-fed infants compared to breastfed infants. So here's some of the statistics that you see that the incidence of NEC in the formula-fed group is much, much higher than in the breastfed group. And the question is, why is that? Do human milk oligosaccharides contribute? Because they're quite different between formula-fed and breastfed. And if so, which oligosaccharide is it? All 150, or is it specific ones? The way we tested that, and we started out about 10 years ago, that we isolated oligosaccharides from human milk, from pooled human milk, to get the total variety of different oligosaccharides, and then tested that in an animal model of necrotizing enterocolitis. And indeed, we found that the survival of the animals is vastly improved. So you can see here, here's your dam-fed control. So that's the animals that stay with mom. These are the animals that we feed with formula. About 25% of them develop NEC and die from it after 4 days. If we add galacto-oligosaccharides to this formula, there is no difference. But if we add human milk oligosaccharides to the formula, you see that the survival rate goes back up almost to the dam-fed control. That is also reflected in histology and in pathology. So you can see here we took slides of the ileum of these animals and then scored them by a pathologist and scored them from 0, which is healthy, to 4 being completely destroyed intestines. And you can see that the dam-fed control had almost exclusively healthy intestines. The formula-fed animals here had pathologies from 0 to all the way up to 4. So it really ranges quite a bit. The only difference between these two groups here, formula-fed and HMOs, is that we add human milk oligosaccharides to that formula. And you see how the pathology scores really drop to almost baseline controls. So that goes hand in hand with the improved survival. Now, that is all 150 different oligosaccharides combined, but which oligosaccharides are responsible for it now? So we used the same model and used a multidimensional chromatography approach to separate oligosaccharides first by charge and then by size, and then we identified that there is this one oligosaccharide that is responsible for this beneficial effect. We then looked at what is this oligosaccharide. And it turned out the structure looks like this, called DSLNT, and that stands for disiallyl, so 2 sialic acids, lacto and tetrose, which is the backbone here, the LNT backbone. So that's the structure that we think is beneficial in the animal model, but it's an animal model. So how does this translate really to humans? We looked around the different compounds here. That's all published, but really we wanted to translate that to humans. And the way we did that is we designed a mother-infant pair cohort where we recruited moms and their very low birth weight infants in different NICUs around North America, and then collected milk samples every 2 days of what went into those infants. So it's the milk that went into the infant on that day. We collected those milk samples, or a small aliquot of it, and then analyzed that for oligosaccharide composition. And then we did some modeling to show is there any associations between specific oligosaccharides and infants developing NAC. And to our great surprise, we found that there was a match. So the same oligosaccharide that we found to be protective in the animal model was much lower in the milk that was given to the babies that later developed NAC. So there's a good match between what we found in the animal model and what we found in the cohort afterwards. That gives us bigger confidence to do a clinical intervention study with this specific oligosaccharide, but also it tells us that the animal model is not as bad as we think usually, and that allows us to really study the mechanism and see how does this all work, what is this oligosaccharide doing, which might give us a clue how NAC actually develops in the first place, which we still have a struggle identifying. So from this example, we found that HMO effect is highly structure specific. In fact, if we remove the sialic acid from the oligosaccharides, this is seen down here, the effect is completely abolished. If you remove only one sialic acid from the oligosaccharide, the effect is gone as well. So it needs to be this oligosaccharide or none. And that is also reflected in the data that we found in the mother-infant cohort, where DSLNT was highly associated with NAC risk, but the other oligosaccharides weren't. So that's one example for immediate benefits of oligosaccharides, in this case for the preterm infant with NAC, and that's highly structure specific. I'll give you one more example here on food sensitization and allergy. So that's exposure early in life, but then we're looking at food sensitization and allergy later on in life. And we published that about last year, and we found to our surprise that there is no significant difference between individual oligosaccharides in the groups that marked for food sensitization or not. So there's no significant difference in individual oligosaccharides. But when we did cluster analyses, we found that there is a pattern of different oligosaccharides, ratios of oligosaccharides to each other that can predict whether an infant later becomes or has food sensitization or not, and that's a proxy for developing food allergies later in life. So what have we learned so far from those 2 examples and many other examples that the time is too short to explain? So sometimes you need specific individual oligosaccharides to be effective. And that's the example of necrotizing enterocolitis. The effects are highly structure specific, they're dose dependent, and the mechanisms are likely receptor mediated. So there's something on the host in the infant gut, in the infant immune system, or on different pathogens or microbes in the intestine that are triggered by this specific oligosaccharide. If you change the oligosaccharide, the receptor will no longer recognize it, and it doesn't work anymore. In other cases, you need specific mixtures or composition profiles to be effective. Here, the ratio of different oligosaccharides to each other is what matters. It's not individual oligosaccharides. And those mechanisms are likely indirect through shaping microbial communities, shaping microbial metabolites, or the entire immune system that gets drifted in one side or the other. So now the question is, what is the optimal human milk oligosaccharide composition? I told you earlier that we're trying to identify what maternal factors drive human milk oligosaccharide composition, but drive to where? Like what's the target? Where do we want to have that optimal human milk oligosaccharide composition? And of course, coming from a formula company perspective, they of course want to know what oligosaccharides do we add in what ratio to be as close to human milk as possible. And I would argue and rephrase that question and say, is there an optimal human milk oligosaccharide composition? is there an average human milk oligosaccharide composition? And I would say probably not. It highly depends on the infant, on infant genetics. Different infants need different oligosaccharides probably. It depends on time. I showed you that the oligosaccharides change over the course of lactation, and they're very different between different moms. They're also different based on environment. So whether you are a baby in Africa or you're a baby in South America, you might need different oligosaccharides. And so that brings it to the geographic location. So I would argue that there is a personalized, context-dependent composition of oligosaccharides, and that's probably true for many other components in human milk. And that's why it's so important that babies are fed by their moms and not receive any substitutes. So in conclusion, I showed you that human milk oligosaccharides are the third most abundant component of human milk. Maternal, genetic, and environmental factors that drive the variation in HMO composition is not fully understood. We need a combination of preclinical, cohort, and clinical studies to fully understand and assess the effects, functions, and potential claims that we associate to oligosaccharides. Sometimes we need individual oligosaccharides to be effective, highly structure-specific. Sometimes we need different mixtures of different ratios of oligosaccharides to each other to be effective. HMOs can benefit the breastfed infant, potentially even the fetus. They may have potential long-term effects, and this is this entire concept of developmental origins of health and disease. HMOs may also benefit maternal health, and they may serve as templates for novel therapeutics way beyond the first 1,000 days. I haven't shown that in this presentation, but we have good data to showcase how individual oligosaccharides might actually help for patients with arthritis or atherosclerosis or other diseases that are more relevant to the elderly. So with that, I thank you for participating, and take questions if there are any.
Thank you so much. That was really informative. I've gotten a question from Dr. Lori Feldman-Winter, who works with us a lot with our CHAMPS project. If one looks at HMOs as the independent variable, how do differences result in changes to the microbiome, disease states, and other health outcomes, particularly with reference to geographic differences?
Mm-hmm. Yeah, so that's the big question, right? How do oligosaccharides actually shape microbial communities, and how does that relate to infant outcome? We don't have a full answer on that. We know that different oligosaccharide compositions lead to different composition of microbial communities. And we also know that different microbial communities are associated with infant outcomes and, of course, the diseases later on in life. There's an interesting study that came out in later last year that showed that the most prominent driving factor of the infant microbiome is breastfeeding. So the biggest variation that can be explained is through breastfeeding. And we think that oligosaccharides play a very dominant role in that. But the data is still needs to be confirmed, and it needs to be studied in greater detail.
Great. Does anybody else have questions?
This is Anne. Can you hear me?
Yes.
I can hear you.
Hi, Lars. I have a question. It sounds a little bit stupid question, but I just wanted to ask you something. When you were talking about HMOs can affect the fetus and the pregnant mother, are those the HMOs that are being made in the mother's body at that time, or is there, are they things that the mother herself has had when she was a baby and she was breastfeeding? Are these HMOs present in the uterine fluids or whatever? Where are those coming from when people are when the fetus, for example, is being impacted?
Yeah, that's a great question, Anne. And whenever we came up with this data that it's already found in amniotic fluid, that was the next question: where are they coming from, right? And we have data to show that this is not just in amniotic fluid at term, at delivery, but very early on. So the second half of pregnancy, you probably find these oligosaccharides in amniotic fluids. And the question is, where are they coming from? Right? Are they really coming from the mammary gland already that are made there and then get into the circulation and then somehow get into amniotic fluid? Well, is the mammary gland already active in a sense that they make oligosaccharides? We don't know that, but we've seen that, not just us. I mean, this is data that's already been available since the 70s, that moms at the end of the first trimester start to excrete these oligosaccharides with the urine. So we don't know where exactly they're coming from. Is there another tissue that makes them? Are they made in the placenta potentially? We don't know. So that's a huge, huge question right now.
Interesting. Thank you.
Mm-hmm.
Does anyone else have a question? At this point, you are welcome to speak up, but you can also type out your questions as well.
I just want to thank you, Lars. That was just really interesting. You know, most of the webinars we have are on very practical things about, you know, how to increase breastfeeding rates and stuff, but this really gives in-depth context to why this is so important, and also how, we hear about these health benefits. We read about them from a popular article to one paper on something very specific. But I think this just informs so much of that. And, you know, when we say, is it really possible that human milk does this? This gives us some clue into the depth of what we're basically dealing with when we start to talk about benefits of human milk. It's really interesting. I do encourage people to speak up if you have questions. I know this is an unusual topic for us, and to many of us it's new, including me, except I've heard Lars before, but the field is new. So please, if you have questions, speak up.
Maybe while people are coming up with questions, maybe to add to that, it is very relevant, because that's what's happening in infant formulas right now, right? I mean, you see the marketing for individual human milk oligosaccharides, so I think it's good to be able to talk about it. What are these things actually, and how similar are they, and what are we doing to our kids there? At the same time, maybe give another example. I talked about the anti-adhesive effects and antimicrobial effects, and we just found an example, and we just published that a couple of months ago, that there is certain pathogens, rotavirus specifically in India, that starts to take advantage of some of those oligosaccharides and increases infectivity because of some of those oligosaccharides. So having that knowledge and studying milk components will potentially protect us from some pathogens taking advantage of us, and we can use that knowledge to develop new vaccines, for example. So this is not just an exercise of looking at chemical structures that we have in human milk. This really has public health relevance if some of those structures are exploited by pathogens and will eventually kill us. So having that knowledge will be highly relevant.
Yeah,
I have another
great.
question. How does milk expression versus direct breastfeeding change HMOs in milk, infants, and mothers?
Mm-hmm. Yeah, that's a very good question. There was just a paper published up from Megan Azad's group that shows that infant outcome is potentially different whether you have direct breastfeeding or whether you express, store, and then feed that same milk to an infant. There's, of course, changes in microbial communities when you store in some of the substructures. We haven't really seen differences in oligosaccharides. Oligosaccharides are fairly stable no matter what you do to them. So if you take this one step further and think about donor milk that gets pasteurized, even that doesn't do anything to the oligosaccharides. They are very stable, and that doesn't change. So we haven't really seen any differences in whether there is direct breastfeeding or the milk is stored and then fed through a bottle. Oligosaccharides seem to be fairly inert to that.
Taking that a little bit further, Lars, given that you've seen some differences geographically and possibly by genetic, I mean, does it raise the question about donor milk or, different milk coming from different women that's all mixed together? Like, does it matter?
Yeah, we don't know that question either, or the answer to that question either. It's a great question. I think the entire field is really affected by that. It's not just the oligosaccharides. Is it important that there is a match that the infant actually gets the oligosaccharides from his own mother, right? And again, thinking of donor milk, most of the donor milk that we use is pooled, so it's multiple moms together, so you're kind of averaging out oligosaccharide composition. But for example, if there is an infant that's supposed to get milk from a mom that's a non-secretor, there's entire oligosaccharides missing, and now it gets donor milk from secretor mom, does that have any impact on the infant? We don't know, but that's great questions, things that we need to study.
That's great. Does anybody else want to speak up or have a question in our last few minutes? All right. Well, if nobody else does, then I think we'll begin to wrap up. But this is your last chance if you have a burning question. If you leave our webinar today and something occurs to you that you really want to know the answer to, you are welcome to email us, and we'll try to get back in touch with you.
Yeah, you can also email me directly. The email address is still up on the screen if you can see it.
Thank you.
All right. Thank you, everybody. I hope you have a great rest of your day. And thank you again, Lars.
Thank you very much, Lars. Thank you all.
Thank you.
Okay,
Thanks
bye-bye.
for having me.